Excitation-Contraction Coupling by Amanda Jones. Everything you do requires muscles. Eating, playing, even something as small as scratching an itch. The process of moving and using those muscles in your body is very complicated. First, it starts with a signal that travels from your brain, that travels down a neuron, that branches when it gets close to the muscle. The signal travels down a neuron to the synapse called a neuromuscular junction. When that neuron sends information or a message down an axon and away from the cell body, it's called an action potential. Excitation-contraction coupling is the connection of that action potential in the sarcolemma and the contraction of a muscle fiber. In a skeletal muscle fiber, the cell membrane is called a sarcolemma. That action potential travels down the neuron and reaches the neuromuscular junction. It then causes the calcium to be let into the axon terminal, or the very end of the neuron. Calcium enters and causes the vesicles filled with acetylcholine, which are sitting in the axon terminal, to be released into the synapse. Then the acetylcholine binds to the nicotinic acetylcholine receptors on the ligand-gated ion channels on the motor end plate. This causes the ligand-gated ion channels to open and release sodium ions in and potassium ions out, causing a depolarization of the muscle fiber. When enough sodium has entered the muscle fiber through the ligand-gated ion channels, it will raise the end plate potential of the motor end plate and cause the action potential to continue traveling. That action potential travels down the transverse tubercles of the sarcolemma to the triad, which is where excitation-contraction coupling occurs. Here's the sarcolemma, here is the transverse tubercles. Here is the sarcoplasmic reticulum. The triad is formed by one transverse tubercle and two adjacent terminal cisternae of the sarcoplasmic reticulum. These terminal cisternae are connected to the transverse tubercles by a combination of dihydropyridine receptors on the T tubules and rhyodine receptor on the terminal cisternae. The dihydropyridine receptors open and allow the release of a small tiny bit of calcium ion. Then that little bit of calcium causes even more calcium to rush through the rhyodine receptors on the terminal cisternae. The next step is the contraction cycle. This is where it gets fun. Those calcium ions that were released bind to the troponin, which is what covers the active site on the actin up here. The active site on the actin is a protein. When calcium binds to the troponin, it causes the tropomyosin to move out of the way of the actin so that the myosin head that comes from the thick filaments can bind to the active site on the actin that comes from the thin filaments. The thick and thin filaments are cytoskeletal filaments that either contain actin, troponin, and tropomyosin, or just myosin. This process of actin and myosin binding causes a cross-bridge to form. ADP, or adenyl diphosphate, and phosphate group, which is PI, are connected to the myosin head on the thick filaments. The PI group falls off when the myosin heads move to connect to the actin. The ADP is lost when the power stroke occurs, which is the actual contraction of a muscle fiber. And what happens when the myosin heads that were bound to the active site on the actin, they get pulled in towards the sarcomere. The sarcomere is the smallest part of the muscle fiber that can contract and get smaller. It's important to know that the individual myofilaments don't actually change length. They just have an overlap, causing it to appear shorter, to actually get shorter. To cause the muscle to stop contracting, we need ATP, or high energy, to come in and replace the ADP and make the myosin head detach from the actin binding site, which ends the contraction. The ADP is then broken back down into adenyl ADP, which is adenodiphosphate, and the PI group, which is the phosphate group. Then everything goes back to regular. Finally, the calcium that was attached to the actin detaches and the calcium goes back into the sarcoplasmic reticulum and your muscle relaxes. If the calcium had stayed, the contraction would have kept going. All of this happens in microseconds, which is super fast, like lightning. Just think about how fast you can snap your finger. This is contraction, and to be able to snap your finger fast, you have to be able to contract your muscles fast. This whole process is how the action potential, or the signal from your brain, and the contraction of a muscle fiber happens. Now, whenever you eat your breakfast, brush your teeth, or scratch that itch, you'll always remember that those small actions are actually much more complex. All of this is further proof that God has fearfully and wonderfully made each one of us. Here are my references.